what we're specifically doing as part of that work is looking at the biomass in straw to see what we could do to try and improve it, to make it easier to make fuels from. So if we look at straw here, actually once you take the grain off a plant, the rest of the plant, when it's dried like this, is really just plant cell walls. The cells themselves are no longer living, they've shriveled up and all the contents have really been used up. And really you're just looking at an empty box of plant cell walls that make up the body. However, in those plant cell walls, there's probably as much sugar as there is in the grain that was taken off the plant at harvest. And that sugar is in the form of cellulose. Now cellulose, what we make paper from, cellulose is really a compound that's made up of chains of sugar molecules joined together in linear chains. And then a whole series of those linear chains kind of bind together into a fibril, a bit like all these stems here in the barley put together, to make a really tough, strong fibril that gives the cell wall some of its integrity and strength. But when the plant gets to this stage, so that it's got really tough and old, or in wood that we use to make a lot of our furniture and things, those cellulose fibrils are encrusted in another polymer that the cell wall makes, and it's called lignin, and that's really the polymer that I work on. So lignin is really, if you think of the cellulose in the plant cell wall in a way like the steel rods, say, in a piece of reinforced concrete, you can think of the lignin, you can think of the lignin as the cement that then goes in and coats all those steel rods and glues the whole structure together and makes it rigid, makes it waterproof, and really makes the whole thing hang together. So if you can imagine then, if you're trying to get at the sugars that are locked up in cellulose within a compound structure like that, it's really quite difficult. And you have to try and break down the lignin and get through the lignin to get to the cellulose, to allow enzymes to begin to break down the cellulose into individual sugars that microbes can use. And so that's really what our research is looking at. So we're trying to understand how the plant makes lignin. We're trying to discover all of the genes that might be involved in making lignin. And we're trying to work out how you could manipulate the amount and the composition of lignin in plants to make it a little bit easier to use biomass to make things like fuels and useful chemicals. And we know from looking at nature that there's huge potential for manipulating lignin, because the amount of lignin and the composition of lignin in a plant varies quite a lot from one kind of plant to another. And even within barley, we've done screens of 850 different cultivars of barley that are grown by farmers. So these are all good field varieties of barley. And even within those good field varieties of barley, the amount of lignin in the straw can vary from about 14–15% to up to 21 or 22%. So the plant doesn't need an absolute, specific amount of lignin in order to stand tall and to be healthy. And the kind of components in the lignin can also vary. So there's plenty of reason to think that this is a trait, a plastic trait in plants that we could manipulate by conventional breeding to come up with a barley crop or a wheat crop that whose straw would be easier to use for biofuel production. So that's what a lot of the research in my lab has been involved with over the last few years. And because it's actually much easier in breeding to work on the basis of what the genes in a plant do, it's easier to understand what the composition of that plant is going to be like when it's grown by understanding its molecular makeup, understanding what genes it has and what specific variants of genes it has, than it is to have to continually do lignin analysis on the straw, grow the plant to maturity and do lignin analysis. We've really been taking a genetic approach where we've been trying to identify all of the genes and gene variants that are important in barley straw for these characteristics. Okay, so I'm not going to go too in depth into the whole genetics of this. What we've done is we've taken these 850 cultivars that I mentioned to you. We've measured the lignin content and the lignin composition in all of them. We've also used an assay to help us to understand how easy it is to get sugars out of them. And this was an assay that was done for us at York University, where they have some liquid handling robots where they can just process all of the material quite quickly and easily. And this is a massive undertaking, because I think they've done something like 40,000 different assays for us over the last few years. And then we've also looked at the genotype, the genes that are present in all of those varieties. And now we're trying to correlate all of that information together to try to understand which genes and which variants of genes are important to make it easier to get sugar out of the plant or to change the quality and quantity of lignin. And down the line, we hope that that information will be useful for helping us to breed better varieties of barley, or indeed any cereal or any straw crop, possibly even trees, with the lignin that makes it easier to extract and easier to use for biofuel and for other chemicals.